Okay, good afternoon. So it's a pleasure to be the moderator of this panel. So we're beginning now this panel about dynamic of organic matter and also soil biology. In my opinion, and I'm a little suspected to say it, but the biological component is one of our key in the no-till system, and it is what we need to enhance and is our challenge, I think, for the future. So we have a good experience from Mr. Amado here in Brazil, and Mr. Wall will bring us a little about the world of the organisms in the soil, I hope. So you both have 20 minutes for your exposure, and after so we will have some time for questions, okay? Telmo, please.
Good afternoon. It's a pleasure to have this space. And I will talk more about the dynamic of organic matter and its relation with conservation agriculture and crop yields. Well, so many thanks for the World Congress, for Brazilian Association of No-Till, and FAO. This presentation is prepared together with Professor Cimélio Bayer. Dr. Bayer is a very productive scientist of Rio Grande do Sul, Federal University of Rio Grande do Sul. He has been many times in Argentina. And with Professor Charles Rice, is a very good friend from Argentina. He's a professor at Kansas State University. Well, one thing that is called attention about the Brazilian agriculture is the big growth in production that we have in last 15 years. I bring some numbers for you. I just checked this morning the last release of our national statistic project that our last season we reached 240 million tons. And to give you an idea about that, our crop production in last 17 years doubled. So that is I believe that was one of the biggest growth in the world. And compared with the last year, there was an increase in 30% related to last year. So it's really impressive, these numbers. And the good things about that this growth is mostly based on conservation ag. Talking about Brazil, is very difficult because it's a big country. We have South Brazil, where one of my friends, I'm see here, Jucaçá, and me, we are from South Brazil. We have in yellow the Cerrado, the Midwest, where is the biggest growth in areas, and is a tropical soil. Well, our experiments about soil organic matter dynamic is 30 to 35 years old. We learned in the last 50 years that when we till the soil in tropical and subtropical environment, we can get a very fast depleted of soil organic matter. In South Brazil, to reach 50% of loss of soil organic matter in 0 to 20 centimeters could take 10 to 20 years, and in tropical Cerrado could take 5 to 10 years. So in more warm environment, the decline of soil organic matter under conventional till is much faster, and Dr. Sa published very good articles about that. We have two long-term experiments, or three, but I'll talk this afternoon about two. One is this that Dr. Simelio Bayer is involved, is a 35 years old experiment. He has conventional and no-till system, and the corn is the main crop. The other experiment that I'm talking about is one experiment that is carried out in Cruz Alta. And this experiment also compares conventional till with no-till, but the difference is the soybean is the main crop. So I try to show some results of these two situations. This experiment is also 30 years old. So the link of soil organic matter and the biology is the input of carbon through the photosynthesis process and the decrease of soil tillage. So the soil distribution need to be associated with high input of carbon. And after that, we see an increase in the biological activity and increase in soil quality. So based on that, in the experiment of URGS that has cover crops during the winter and corn during the summer, after some decades, we saw the increase of soil organic matter, and let me see. Well, and this was associated to the carbon input. After some time, we saw that no-till is going up, the carbon, and conventional tillage with low input carbon go down. Even this experiment from URGS and the experiment from Cruz Alta, they didn't reach what we call carbon saturation until now. Maybe if you keep going with this experiment for 50 to 75 years, maybe we reach this carbon saturation point. One question about the carbon loss was the erosion. We didn't count how much of carbon was lost by erosion in this process. Let me show some data. This year, in May, during 2 months, we got 951 millimeters. It's the half of the precipitation that we have in the year we got in just 2 months. And in 2 days, we got 263 millimeters of rain. So the issue about erosion is still in our agenda. We still have a lot of erosion when we didn't protect the soil as this farmer did here. And I start my presentation talking about soybean. And soybean, the residues of soybean is very light, and so it's very easy to get soil erosion after soybean. So we need to protect our fields with cover crops just after the soybean, and we try to see that with our students. But the long-term experiment of URGS showed to us that under conventional tillage, any system of crop system was able to get some gain of carbon. The more intensive carbon input system that we have was just able to get a stable carbon amount. And no-till, with high input, we got increase, a significant increase in carbon. So it's showing here that conventional tillage with low input of residues, we have a big loss of CO2. No-till with low amount of residues, very low impact. And with, we have, in case of corn, legume cover crop, we got an increase in corn. And also in this experiment, they try to look in the new and the old carbon, and we see that this crop system was increased, the total organic carbon, and even the young carbon is increased the percentage in relation to the old carbon, and that's not happened when we have conventional tillage with low input. And there is a relationship that was observed also in Paraná, in other states, for Calegari and Juca, that the amount of carbon that we are able to input was directly related with the amount of carbon in the soil. But the tropical soil, the oxy soil, has some special issues that we should consider, and I would like to address now. One important thing is when we have soybean in the system, usually we got the lowest increase in soil organic matter. When we have corn and other systems, we got higher. So we need to change that. The experiment of Cruz Alta shows very clear that here is no till with soybean and wheat rotation, and the increase in carbon was very little. And the highest increase with soybean was when we was able to introduce a crop system with rotation. The reason for that is related to soybean carbon footprint. This is in this experiment of long term that we are able to put an eddy covariance tower, and we are able to measure the amount of carbon that is released and entering the system every 15 seconds. The result that we got was that the balance of carbon of soybean, when we take the carbon that goes out with the grain, was negative. Or what can I say, that soybean will be not increase the carbon in our system. We need to go for another crop system that could be associated. Well, we have some crops that we can associate. This is brachiaria, and we see the amount of roots that we can work. The idea to work with root system to build up soil organic matter is very important to tropical environment where the above ground biomass decomposition is very fast. So this is the brachiaria consortiated with corn, and after that we have soybean. In this system, the buildup of soil organic matter is going very well in central Brazil. But what I'm going to show now, that in tropical oxy soil we have some challenges. One challenge is the soil compaction. The soil compaction decreases the root growth, and that's a problem for us because we are we need deep root growth to build up soil organic matter. The other limiting factor to root growth is the aluminum. The toxicity of aluminum that is very common in tropical soils, and the calcium. The lack of calcium in depth. So it's very common, even in crops that have been in production for a long term, that the amelioration of the soil with the lime applied on surface under long-term no-till, that we create a shallow layer where the roots concentrate, and in subsoil below the 20 centimeters, we have low calcium and high aluminum. And we need to change that without disturb the soil. So this is a challenge for oxy soils. I show some data, some of our data. with gypsum and lime. You see here only lime applied on surface, and here when combine lime and gypsum, and we got some of this calcium moved down in soil profile. With the calcium moving down, the relation between aluminum and calcium is changed, and the root system go down. This is in Paraguay. 4 weeks ago, I was visit a no-till farm down there. He was applying lime on top of brachiaria. And there is some interesting things here. He was applying with the best tech of precision ag. He was using RTK signal and was applied with equipment that has a very good distribution. But the question was, why they need to apply lime on top of brachiaria? And he explained to me that he wanted that the decomposition of brachiaria release organic components and help calcium to move in the soil profile. Also, the roots of brachiaria, after decomposition, create a porosity to some lime go down in the soil profile. And that is the same that we have in Paraná data some years ago with Pavan and Miyasawa that showed the lime applied without black oat residues. The correction of soil profile is going only to 10 centimeters. And when we got crop residues of black oat, this correction is going much deeper. So this farmer is sort out his problem this way. Another way to deal with that, or is another point of no-till, is the phosphorus. Phosphorus is a key nutrient to stimulate the root growth. And if we apply the phosphorus deep, we can stimulate the root growth in the soil profile. I show some data. This is one equipment that was able to put the phosphorus 20 centimeters depth. We carry out research regarding this equipment, and the data that I'm showing, that treatment is this one here, and we saw that the root system grows in depth much higher than in the shallow application. I show a picture about that. This is the broadcast shallow phosphorus, and here when we put the phosphorus on depth. So calcium, we talk about calcium, we talk about phosphorus, and this equipment is to sort out the problem of soil compaction, if we have. If we have soil compaction, we need to sort out the problem. One way to do that is using a chisel and associate that with cover crops. This equipment is seeding the cover crops at the same time that is broken the soil compaction. I know the issue is, you can have different views about this, but it's an option. Another innovation that we have is equipments that put the lime on depth in no-till system. This is one of equipment that do that. And, but the important point is associate that with cover crops, or even better than cover crops, a mix of cover crops. So here we have where we got the soil release of soil compaction and the roots of a mix of cover crops to restore the soil quality and increase the soil biology. And here it's how it's look sometimes after. This is another innovation that we are looking for, is associate controlled traffic with cover crops. This is a work that we have been done with one farmer. And here is where all the traffic of machinery was concentrate. He used red soil to promote soil discompaction. And here, where there is no compaction at all, because there was no pass of machinery, he used black oat. And even we can do that following the slope to avoid erosion. But what I would like to finish my presentation, just show that it's possible to conciliate conservation agriculture with high yields, high soybean yields. I show you here two friends mine. This is from Rio Grande do Sul state. His farm reaches 6,100 kilograms of soybean per hectare. That's very good. And this is from Paraná state. This is the farm, the cropland of this gentleman, and he has hair vetch in his fields to bring nitrogen and biological activity. In his field, we made a precision egg earthworm counting, and we count 50 earthworms per square meter. So some places we got a lot, other places less, but that was very good data. This is another example of yield, soybean yield in Rio Grande do Sul state. See, Vanderlei, his father started the no-till, 25 years, he's the son, and he put the disc of plow in his farm, the same that Nono Pereira made, to show, well, we can get very good yields of soybean with following the principles of conservation agriculture and some innovation. Here's the soil of his cropland. Well, my time is flying. I hope that I could show some ideas of innovation in conservation agriculture to conciliate high yields. That is the challenge that we have nowadays. We need high yields to pay our bills in the farm and conciliate the environment issues. Thank you very much.
Thank you, Telmo. Very good data that we can discuss later. And so now I would like to, Mr. Wall, to your talk, please. 20 minutes.
Good afternoon. First of all, I want to thank the organizers and this World Conference on Conservation Agriculture to give me the opportunity to tell some of our recent results in soil biology and agriculture. I was asked to talk about dynamic of soil biology in soils under conservation agriculture, so I will discuss about how soil biology is changed with time, and I will focus in early effects of soil management on soil biology. And when I talk about soil biology, I am referring to everything that is a living part of the soil. Soil is an alive system. Soil is not a structure where things are living. Soil is the result of the living things within the soil. So with this idea, I will focus in two of the main principles of conservation agriculture, how these principles affect soil biology. I will discuss the no-till effect and the crop rotation effect separately. In order to do that, I will start discussing the tillage effect or no-tillage effect. And for that purpose, it is many years I came for the first time to Aapresid, and I found in Aapresid for me, it's like Disney World. It's plenty of essays with farmers that I just want to jump in and to make measurements. This is one case. In the southern west part of Buenos Aires provinces, the group of farmers gather at the regional of Bahía Blanca. They have a very interesting essay, a long-term essay, comparing no-till with the conventional tillage. I was here in a congress in 2012, and I was introduced to this essay. And you know, today in scientists, we are rather particular person. I mean, when we found some system that is organized, we want to break it disorganized to understand why it was organized. So that's what happened with this essay. I was here discussing with Jill Clapperton about that this essay was a perfect baseline to study what happened if we turn one soil management into the other. So I came back to Christian Kleine, the responsible farmer for this essay, and first of all I told him, well, this is a fantastic essay, but I would like to break it a little bit. And he agreed, and we did it in 2013. So part of the plots was just flip-flop the management. Interesting for this essay is that the crop is exactly the same at both sides. So there is no plant effect that we should care about it. It's just the soil management. And we took soil samples by 5 subsamples at each treatment. Now we have the no-till, the conventional tillage at the historical controls, and the new conventional tillage and the new no-tillage. And we took samples 6 months, 18 months, and 30 months afterwards. And there was no change at the organic matter, even at the 30 months after the change of management. But curiously, we took a measurement about the proportion of microaggregates, and we found that the new conventional, the new no-tillage increased the amount of the bigger microaggregates, and in the new conventional tillage was the opposite. This was something physical in the microstructure of the soil, and I suspect it has to do with the biology. I am a biochemist, so we decided to start to look for the ecosystemic services of soil related to cycles of nutrients. So we measure a profile of enzymes that are deal with different cycles of elements, phosphorus, carbon, nitrogen, and sulfur. We measure the enzymatic activities, and with the activities of the whole enzymes, we create a profile. So I will ask your attention to compare. This is the enzymatic profile of the 4 treatments. The historical no-till is the full green line, and the historical conventional tillage is the full red. And this shows that the enzymatic activity of soil and the no-till is bigger than the conventional tillage. And this is 6 months after the change of the management, and the new no-tillage and new conventional tillage show like a different behavior. It was not similar to each other, but not similar to the controls. After 30 months, the new no-till became higher in area, more similar to the historical no-till. And the new conventional tillage reduced in size, becoming similar to the historical conventional tillage. I mean, in the soil functioning, the soil functioning after 30 months start to behave as the present management of soil. Using another way of comparing this profile, we could see that both profiles are different in the historical management, and the new management show a different situation, and after 30 months, they become similar to the historical one. So this was for the biochemical point of view. Then we took DNA from soil and quantified the big groups of microbials, bacteria, fungi, and archaea, and this is how it differentiates the new treatment compared to the original one. And we found that in the new no-till, bacteria, fungi, and archaea, the three groups increased in particular microaggregates of the soil. For me, it was curious to find that archaea, that is a biological group that is always neglected, ignored, it is particular increase in no-till. I have no idea what does it means, but it makes, it's interesting to find that. We should take a look at that group. This is something I would like the last one to please read this. Now, this is not the idea. This is another way of looking at the microbial diversity in soil. We took DNA, we amplified the general gene for bacteria, and we make a massive sequence of that DNA. In that way, it's like to take a look at everything in soil. Not everything, but we are closer to that. And this is the more abundant groups of bacteria. And here we have all the samples we took. What I ask you is to see that here we can see at least two groups. This is one half, and this is another half. And in this half, there is also two kind of half, and in this group, there are two columns. This is, when we send the DNA samples, they are blind. I mean, the one that make the analysis has no idea about what is in the sample. But it makes sense when we put labels, because this is the bacterial diversity of no-till soil, this is of conventional till, and this is the lands that have been changed in soil management. And this is interesting because these two columns is the differences in depth. This is 0.5 centimeters and 5.10. In conventional tillage, that you remove the soil, it's not easy to find the depth differences of bacterial community. So this is a very intriguing result because the soil structure is not yet similar to the historical treatment, but the function was. I mean, the enzymatic activity was. This is one part of the story. The other part is the effect of crop rotation. To look for crop rotation, we take a look at no-till soils, and we work with another group from Aapresid, the Regional Pergamino Colom, that they have started in 2011 an essay. In Aapresid, they always raise the bed. And they start from the conventional typical rotation, and they try to intensify the crop rotation until pasture. Well, I mean, this is an index of the days of the year that is covered with plants. And from a plot that has the same history, they divide it into different 5 treatments, and try these different rotations. I think it doesn't matter to pay attention of the rotation in particular, but they are with different intensification of rotation and different diversification in the crops that make part in the rotation. And after 3 years, only 3 years of this rotation, the farmers, this group of farmers, didn't find differences, significant differences at the chemical and physical level of soil. So they asked, they came to us, and we decided to look into biochemical and biological factors or indicators. This is the, we did as as Marie showed yesterday, we took these blocks, 25 by 25, and sent hand sorting of earthworms. And also we took samples of for mesofauna. There are 6 posters in the room. You can take details of that results. And this is just to show that the density of earthworm only after 3 years of starting the differentiation of crop rotation show in the different fields always a highest value in the intermediate rotation with high percentage of legumes. At least has 2 different legumes in the rotation. And this was particular to have these results. When we took a look at in a multivariate analysis to the whole macrofauna that was detected in those samples, it was clear that the pattern of macrofauna was different between the intense rotation, the pasture, and the typical rotation that was similar to a monocropping neighbor field. Regarding biochemical activities, we measure again enzymatic profiles, and the enzymatic profiles easily distinguish between the different treatments that were the same at the beginning. It was curious for me as a biochemist to find that the activity of the enzymatic enzymes in soil, that means these ecosystemic services regarding to element cycles, increase in a very good correlation with the intensification of rotation. So it makes possible to perhaps to have a reference value for different soils in the future. And we also took samples of soils to extract lipids and to have a lipidic profile of soils, whole lipidic profiles of soils. And again, the lipidic profile differentiates between the rotations. And again, I have not all the responses, but I don't know what they mean, but we have particular fatty acids that seems to characterize the different rotations. Also, the amount of macroaggregates were differentiated between these 2 rotations. So a lot of changes happened at the biological level after 3 years of this change in the intensification of crop rotation. So, regarding conservation of agriculture, I went into the dictionary. I looked what means conservation. Conservation means preservation or restoration from loss, damage, or neglect. So I ask, what is lost, damaged, or neglected? In present agriculture, that as it was told this morning, it's an agriculture that is based on a chemical paradigm, is since the 40s, and is based on the knowledge in the 40s about plant physiology. Plant needs nutrients. Anybody that studied plant physiology probably never listened about microbes. But nowadays, in the present, we know that the plant interacts with a huge amount of microbes. But in the old times, plants only need nutrients. So it's like the soil is like a flower pot. that you can put nutrients and grow the plants. So these ecosystemic services that are provided by the soil biology were replaced by chemicals, by fertilizers, by plaguicides, fungicides, antibiotics, herbicides, and it worked very well, even better than just the soil. But the problem is with the bat. The problem is the impacts in the environment. The problem is that we have now an agriculture that is dependent on chemicals. Now we have weeds that we don't have it before. It's like with your health. If you use chemicals, antibiotics, you have problems with the resistance of antibiotics. It's the same. It's just biology. So I am very proud of giving this talk in this meeting. I think it's fantastic to have this meeting in the context of the Aapresid meeting, because we push all together in order that agriculture takes the biological paradigm into it. I am not asking to replace one by the other, but this is not a romantic hope. With our results and results for many other people, Marie results, we show that it is possible to do intensive and very productive agriculture, not just taking care of soil biology, but improving soil biology. Intensifying crop rotation, we improve macrofauna, mesofauna, and bacterial diversity. So my take-home message is farmers should take into the analysis the ecosystemic services and try to make use of it. My impression is that cover crops will help to do that, because cover crops is helping to improve the soil biology. Just to finish, I want to recognize Luciano Gavarini, Juan Frene, Belén Robledo, Julieta Covelli, Alejandro Ferrari, Bettina Garas, Dalila Reina, y Claudio Valverde, people from my group. Belén Agosti from the group of Chacra Pergamino, Christian Kleine from Fundación Funke in Bahía Blanca, the University of Rio Cuarto, Bedano and co-workers, people from CONICET, and Cornelius Mala from Germany, that they help us with the metagenomic analysis. And just from literature, I got a present from a person I love that gave me this thought that comes from literature, but I think it summarizes very well the spirit of this meeting. Thank you very much.
Thank you, Mr. Wall. I think in my vision, both presentations are completely complementary. Mr. Amado, he showed us that the importance of the time for the type and the quality of the organic matter and how it can enhance our production. And Mr. Wall clearly showed us that the organisms of soil are quite important to enhance this part of organic matter. So if we don't have this whole cycle from biological, plants, animals, microorganisms working together in this system, we will not could do this conservation agriculture in a good way. I think now we can open for some questions. No? No, nenhuma. There's no questions on text. See? No on text. We want to know if someone of the public have any questions. There is one, please.
I think both are excellent talk. I have a question for Dr. Wall. The changes which you see in microbiome or soil biology, have you ever tried to relate it with crop productivity? Because at the end of the day, farmers will be looking at the economics and the productivity. So at what point of time you will see there is a relationship coming up?
Yeah, Thank you for the question. We have not this answer, but I am completely aware of that. Now, in the case of the essays of intensification of crop rotation, we will have the opportunity to test with the same crop the metagenomic analysis of the same crop growing on different histories of intensification of crop rotation. And I guess we will be at least able to detect candidates of markers of productivity of soil. And at least in old time, we were able to use metagenomic to find the putative candidates of markers of activity, and then it's possible to quantify it by qPCR going back to the soil. And we were able to reproduce some results comparing monocropping against crop rotation. But I guess it will be possible. At least it's a working hypothesis.
Any more questions? No? Denizar.
My question is for both speakers. Do you believe in the future we will have a kind of microbiology or biology indicator to substitute the analysis of soil fertility in practice?
Well, what we have nowadays is some sensors that we can follow how the plant is growing. And with these sensors, we are able to detect that the nitrogen, for example, have some deficient, and then we can try to sort out during the cycle, or even sampling the soil in that point and check if this deficiency is also in the soil. So for vigor of plants, we are already advanced. I believe that is a future on this way. But to drive the soil sample, not to don't use. I believe that maybe because we have this issue of spatial variability that is very hard to deal. Even in the soil biology, soil variability is very tough issue to deal. Maybe the kind of sensor or image, satellite image, during the plant growth can help us to locate where the problem is, and then we try to sort out. For example, if you have low biological activity, and that is related with low plant growth, we can go to that place and put organic fertilization or try a specific cover crop that can restore the soil biology. That's something that in precision ag we are discussing if it's possible have multiple cover crops in the field to try to sort out problems. I try to show one example with the controlled traffic that we know that there is a concentrate traffic of machinery, and we use a specific cover crop that was hedge soil to sort out this problem. In big scale, maybe we can select the problem areas and try to sort out a specific solution for that.
I only want to add that I think it's possible in the future to have something like a pregnancy test that the farmers can buy and test the activity of soil for enzymes. The main problem is to have the reference value. And for that, it's necessary to have a big program that means a scientific policy. I mean, to share efforts to characterize soils at different regions between Brazil and Argentina. It's a problem to have the reference value. I think it's not impossible. It's just a matter of investment of efforts, not only money, efforts.
Mm-hmm. Just to complement Mr. Wall, Denisa, in Brazil, the researcher Ieda Mendes, she's doing a kind of enzymatic analysis, and she's trying to do a routine methodology that we can do together when we have a sample of fertility for chemical analysis, also for enzymatic. But that's the problem yet, to do the standards from the different kinds of crop rotation. It's quite different. We are talking about organisms, and that they vary a lot depending the region. Brazil, it's enormous. So talking from earthworms, we have species in 100 kilometers, 50 kilometers, there are different species. And we still don't know what we have already. So depending the organisms that we are talking, we have already a lot of work, but I think that it will be possible soon, we hope. As you see, he showed his team for work. The teamwork for soil biology and for research, they are quite big. The work is not hard. It's quite a simple work. But we need a lot of students and people helping in this work because it's trabalhoso, yeah, the stuff to do. Yes, from coordination, yeah.
And to put all the efforts in the same sample.
Yes, that's the fact. But we are working in that, and Mr. Wall. Thank you.
Yeah,
so.
But I agree that the soil fertility, especially in croplands, has been fertilized for a couple of years. Is limited in relation to express the soil productive potential. And then we need these tools, biological tools, to understand the soil quality, the soil productivity and soil function. And so it's clear to us that we need a more holistic approach to understand the soil quality and the soil productivity.
And maybe I think I pointed the farmers need to have in their head is that soil biology, you cannot enhance it from one year to other. Mr. Wall was very clear with that. Soil biology organism, we need time to that can grow and be in the system. So at least 3 years that you can see the first results, and you need to invest. And the main resource for soil biology is the crop rotation, is the food that you give, that you put in the soil. So it's the diversification of food that you will have a diversification of organisms in the soil.
And the nice point that we can explore that. And besides use the cover crop, we can associate physical and chemical strategies to get more growth of cover crops during the time that we have to restore the soil quality and enhance the soil biology. So I think that some farmers are doing that, are harvesting very good results, and we should pay attention on that.
Any more questions? Gottlieb? Is there how much time yet? One more? It's the first.
Thank you. Today we know that there are many approaches and attempts to evaluate soil quality and to advise farmers whether they should do something about soil quality. So 2 concrete questions from a practical point of view. First, regarding soil organic matter. Do you have any idea, or are you working on thresholds regarding the levels of soil organic matter up to which a farmer should actually insist or invest in improving soil organic matter? And the other one is, what parameters, what indicators would you suggest for a farmer to look at in order to see whether his soil biology is good, bad, in the middle, or whether he should really do something about it? Is it just the count of earthworms? Is it something else? Thank you.
I think in terms of practical terms of soil biology, it is possible to have some data. I mean, what I show is biochemistry can show farmer if the management is going in the right way or in the wrong way, before maybe before the physical structure is changed or the organic matter has increased significantly. That's something I was taught by farmers, because when they were enthusiastic about our results, I thought I was doing basic science, and they told me, no, this is what we need to see if all the effort we are putting to improve soil quality is right or wrong. So I think it is possible. Then it's again the problem of the reference, the value of reference. But that is, I think it's like in clinical. It took a long time. You know what is a good cholesterol value for your health. Well, and now people know what is the expected or desired value of organic matter. Well, we'll learn in the future about the values of activities of soil or groups of particular microbes. I think we are in the process of developing knowledge, but I think we should do, because without knowledge we will not do anything.
Well, the first indicator of long-term experiments showed to us in subtropical environment, in tropical environment, we should have an input of around 8 to 10 tons per hectare of above ground biomass to keep the soil organic matter level. Then, with the advance of soybean, this amount of above ground biomass input changed to be very hard to reach. So the idea was work more with the root systems. Try to use the the windows that we have to have a crop with a very good root system that could compensate the amount of above ground biomass that we are not able to reach. And by add roots, we can build up faster the soil organic matter. We can stimulate the biological activity and soil aggregation that you show the macro aggregates that we observe at the same time. To get a higher root growth, we should overcome the limitation factors, and that is for each soil is different. For oxy soils is the calcium, low calcium, high aluminum, low base saturation, and sometimes the soil compaction. So what we try to show that with some innovation we can overcome this problem of stratification. Because if the soil fertility is stratified on topsoil, the crop residue is added on topsoil, everything goes just on the skin. And to build up the soil structure and the soil quality, we need to go deeper. And I believe that the root system is a driver for that, because with the exudates, we have the soil biological activity. And so I think that we are looking for some new indicators that we can capture that in practical way. But we have, at least in Brazil, many farmers now sampling in depth the soil, try to see how is the stratification, and open, dig walls and see how is the root system. If the root system is too shallow, that is an indicator that the soil quality will be not good. So we try to have a better root system distribution in the soil profile as an indicator of soil quality, soil biology, a practical indicator.
Can I complement it? In Brazil also, we with this project Solo Vivo from Embrapa that we are working in 6 different states. And in one of the meetings, we have had about 40 researchers together, and it's not easy to have so much researchers together on agree and some things. But in that meeting, we really agree that one factor, one indicator it's something that a farmer can see and it's easy, and that's really the soil structure. And we kept this as some Amanda Nunes, and we have some posters, and then we began, they are based on the VSA of FAO and VES, another methodology, and they do the DRESS for Brazil, is to see how is the soil structure in the soil. And of course, if you have a natural soil, that is our reference. If you have a native vegetation with native soil, and you compare with what you have in your field, and the farmer can easily see how is his system working in that way. And that is something also from conservation agriculture. It's not only for laboratory analysis. That's the point that a farmer, he needs to look his soil, he needs to open the soil to dig some holes and feel what is happening in his farm. That is something at that point also in conservation agriculture.
No more time.
Finish. If anyone have any questions, I think the speakers will be available later. I will thank you, the public, for being here with us until now, and also a special thanks for the speakers. It was really great. So thank you very much.